disjointed, probably difficult to follow. So what I'd like to do today is kind of go back through some of the same information, but more in a structured way. There was a request to cover more of the regulatory environment and how that fits into this whole process, as well as intellectual property. So I'll cover that today as well at a high level. And this will be important in doing your projects in that as you identify companies, you'll get a feel for what it is they've accomplished, what is it that they really have, but maybe more importantly, what's ahead of them and what do they need to accomplish over the next 3 to 5 years to make it a good investment. So this is really about kind of building more of that content knowledge around the drug development process. This presentation is a little bit more focused on small molecules because that's more of my background, but I'll make the bridges to biologics where I can. But a lot of the information is applicable to antibody companies, companies working in the protein space. So the next slide. Let's just go right to the next one. So in terms of the drug development process at a really high level, you'll start with your early drug discovery work. Once you start getting some leads out of that, then you have a process called lead optimization, and that's where you've got an interesting compound, and now you're going to optimize that compound and try to get to a real drug candidate. Once you believe you have that drug candidate that you think you can take into clinical trials, that'll kick off your IND-enabling toxicology package. And so this is really your tox work to get an idea on safety and safety margins prior to going into clinical studies. You will then file your IND, your investigational new drug application. That will then give you permission to start clinical trials. If all of that goes successfully, you'll file your NDA for a small molecule, new drug application. And I'm going to go through each one of these in detail. Or your BLA. The FDA will review that, then there will be an approval, and here I have post-approval activities, which is really phase 4 studies, commercialization, all that stuff. But that's outside of the scope of what this presentation is for. So I'm really going to focus on early drug discovery to approval. And then next one. So moving into those research activities. So next one. So really at the beginning of early drug discovery, in small molecules, you're looking to discover new chemical compounds. Just up here is an example of Vertex's Telaprevir, which is a very successful HCV protease inhibitor launched last year. In looking at small molecules, there's really kind of two main approaches that companies take. One is rational drug design, so this is a very targeted approach, leveraging crystallography, and the other is high throughput screening. So in rational drug design, you have a target. You're very interested in that target, you do a lot of crystallography on that target, and you're looking to design a compound that interacts specifically with that target. High throughput screening is very different. This is more of an automated process where you have libraries of compounds, tens of thousands of compounds, and you're just screening them, looking for a hit in the assays, and if you get that hit, then you'll kind of start drilling down on that a little bit further. This is an approach that's used commonly in large pharmaceutical companies, not something that's usually used in a small biotech because it really requires a lot of mass and scale. And so as you're working in that new area, you get an initial characterization, you get some compounds, you like those attributes, and then that becomes your lead. And this compound is not your drug. It is a lead, and then you're going to take that into the next stage, which is lead optimization. So you've discovered this compound, and now you're going to start optimizing that compound. And what you're doing here is you're looking to obtain a compound that can be your drug candidate. And so your pharmacodynamics, you want good PD, how does this drug bring about its characteristic effects? So here you're looking a lot about efficacy and potency. Your pharmacokinetics, your ADME, so your absorption, distribution, metabolism, and excretion. You're looking at safety, and you're looking also at your physical chemical properties. And here you're really focusing on, is this a drug that you can manufacture? Can you manufacture it at scale? Will it have stability, solubility? So you're really looking for the good drug-like properties. And in this sense, you might be screening thousands of compounds. And so what you may end up doing is you could have what we would call a screening cascade, maybe a year's worth of studies leading from an initial look at potency all the way to, and I'll get into this concept a little bit later, non-GLP toxicology. So you start doing a lot of more expensive, longer-term animal studies. And that whole cascade may take a year to get through. And what your medicinal chemists are doing are they are just cranking out compounds. following along a certain structure, so following along your lead, and then putting those compounds through your screening test. Start designing around those metabolites. Try to create compounds that don't create those metabolites. And this is a very long process. And as I said, it can start with very simple in vitro and in vivo experiments where you're trying to rule out compounds quickly because you're pushing a lot of them through. Start doing some PK studies in rodents. Does that look good? Then you start moving into some in vitro and in vivo, probably non-GLP safety studies. And at the same time, your CMC folks, which are your manufacturing folks, are starting to see, can we scale this process? Can we really make this compound? And as I said, you're putting hundreds of compounds through, and you're really looking for that drug candidate. And this can be a pretty extensive process. Next slide, please. And once you've gone through that lead optimization process, what you really have is you have a compound that you feel can be a drug. And what you're going to do from there is you're going to start your IND-enabling toxicology studies. And so your IND is your investigational new drug application. This is the application you're going to take to the FDA for permission to start clinical trials in humans. And really what you're doing there is you want to have a compound that you think can make it through. And so at this point, you're going to start your non-clinical toxicology studies. And so this is really about getting an understanding of the safety profile of the drug in animals. And usually you kind of pick two species. Typically it's rats or mice are going to be one of the species and dogs is the other. And you're looking for toxicology, and safety margins. And so really what you have is, before you start this, you're going to have some idea of what you think your therapeutic dose in humans is going to be. And you're going to try to bridge that to the animals, and you want to get an idea of how much of a margin are you going to have for that compound. And if you don't have a margin, or that margin is very narrow, it's probably going to kill that compound. You're going to need a big window. And the toxicology, the pharmacology, and the PK, these are all interrelated. And the initial dose that you're going to propose to the FDA is going to be based on these IND-enabling toxicology studies. And what you're going to be doing is it's a process that you build upon. So the first thing you do is you look at single-dose studies, typically in 2 species, but a lot of times you'll do it in more because you don't quite know how the compound's going to work. You don't know exactly what species are going to be the best. And so you might do single-dose studies in rodents, primates, mini pigs, all kinds of different toxicology models. You may be looking at genotoxicity, and then you'll move up to repeat dose studies and start building that safety profile for the compound. And going from lead optimization, so that time you start getting a hit and you start thinking about, do I have a drug and how am I going to optimize that compound, through your IND-enabling toxicology studies can take up to 3 years. Your IND-enabling toxicology studies will usually take about a year or so. And so you've got a couple of years of work on the front end of that before you identify that clinical candidate. And then it'll take you usually about a year to complete your IND-enabling toxicology studies. And so all of that takes several $1 million. And in the background of all these studies, you're also doing what we call our CMC, our chemistry, manufacturing, and controls work, but you're doing all your CMC work so you can have a drug product or a drug that you can take into clinical studies. I'll talk a little bit more about that. And so the IND, it's basically what you file as a request to the FDA for permission to use your experimental drug in patients or healthy human volunteers. It deals with all your preclinical findings, so all the animal studies, all the in vitro studies, all those studies that you've been doing are part of that package. Also, your method of manufacturing the drug are also part of that. So it's not just the animal work and the lab work, it's also all the work dealing with the manufacturing of the product, as well as your proposed protocols for those initial clinical studies. It's important to note that these preclinical studies are done under what we refer to as GLP, good laboratory practices. So these are not just kind of researchy type studies. Some of that stuff you'll do in the beginning where you're trying to select compounds, but the package and the data that's going to go before the FDA has to be done under GLP. Also, your manufacturing has to be done under GMP, which is good manufacturing practices. So in the beginning, you're making the compounds at lab scale. They can be a little dirty. You may not have your process down. But what you submit to the FDA, you've taken it from the lab, you've scaled it up, and you've gone into a GMP qualified facility. And especially for smaller biotechs, you've contracted that out to a contract manufacturing organization, and there's plenty of them out there. but you've made that, you've done process development, and you've made that transfer of the process to a GMP facility, and they are producing material. And with that, you're going to have some amount of stability data as well as some amount of drug product data as well. And so it's more than just the preclinical studies. It's all your preclinical studies, it's all your manufacturing, and it's your proposed protocol. And all of this work is very standardized, it's validated, it's rigorous, it's done by trained professionals. And so it's a very structured process. And prior to filing that IND, you can request a pre-IND meeting with the FDA. This is really your first time to interact with the FDA, get them acquainted with what you're doing, start building a relationship with who they are, and you start getting feedback on the application. You start getting feedback from the FDA on what they feel. And as we talked about last week, sometimes what they feel at one point changes later in time, and so that creates a lot of problems. And these are pretty big, up to 15 volumes, 300 to 400 pages each, so it's a big application that goes in front of the FDA. And so maybe that's kind of real quick through the research, so maybe if there's some questions on that part before I transition.
Just a quick question. Is this a preclinical research trial phase 1 or before phase 1?
This is all prior to phase 1, and so all of this work that you've done, the early drug discovery is to see if you can get a lead. Can I get something that looks interesting? The lead optimization is creating what you think could be the drug. And then the IND enabling toxicology is the safety work you need to do before you can even go into phase 1. So all of this work is done before your clinical trials, the first clinical trial being your phase 1. As I said, this could take several, assuming everything works out well, it'll take several years and millions of dollars just to get there.
I didn't have a huge desert.
Oh, okay. So it's a pretty big process, and so a lot of times when you're looking at small kind of biopharmaceutical companies that have been spun out of universities, this is the type of work that's being done. They'll set up a lab, you'll have 3 to 5 medicinal chemists, you'll have a few folks in biology, and you're cranking through the compounds, you're getting an understanding of your target, and you're building up to filing that IND.
I have a question. So I know a lot of large biopharmaceuticals, especially when they're looking for new drugs, can go through like thousands of potential drugs before they actually come up with one particular one they want to pursue. How is it with smaller startups? They tend to have a much more concentrated group of ideas that they're looking at?
Yeah, so with it, you're limited by how much capital you have. And so the process may be a little bit different. But the idea is still the same. You're trying to get that hit, and then you're trying to characterize that compound well enough to think that you have a drug. And what you'll see with small biotechs is you don't have the luxury of spending a lot of time and a lot of money. So what they'll usually do is they'll get a compound that they feel is good enough. It's safe, it could be better, and you'll push that into clinical studies. And so what you'll have is you'll have your lead compound, and then you'll have what we'll call like a robust backup compound, where you're continually working on it. And when you go to try to do that BD deal with pharma, you're selling them not only on your lead compound and all of its characteristics, but you're also highlighting what you've got in your backup. That you've got, better potency, better, say if you're doing Alzheimer's, better blood-brain barrier penetration. You think you might have a better safety margin. And a lot of times what you'll do, especially in small molecules, is you'll just, even though you have what you think is a clinical candidate, you're continuing to bring compounds forward, and you'll park them in certain places. So you may have a lead that goes into phase 3, but you may bring another compound into phase 2, maybe a couple into phase 1 that have different characteristics. And so you have that backup program that's parked in the right place behind your lead. But that effort in a small biotech is really determined a lot by the capital that you can raise. And you have to get compounds out. You have to create value-creating milestones. You have to try to get a partner. If you're going to have to raise more money, you want to have a more compelling story so you can do it at a higher valuation. And when you're able to raise money at a higher valuation, your existing investors don't get diluted as much. So you're kind of trying to play all those factors in as you're trying to decide, should I move something forward or not? And, what kind of studies am I going to run? Especially some studies, especially primate studies, are very, very expensive. And so you're trying to really optimize all these different considerations. But yeah, it is what a small biotech will do and what big pharma will do is very, very different.
So parallel to this research pipeline, what's the test about for outside help to get equally financial and legal help?
So the legal help is going on with this. So it depends on, if we're talking about a small biotech, you know, you have two kinds of legal help. One is your corporate counsel, who's helping you get all your financing docs together and making sure you got good NDAs in place and making sure, you know, you've got good consulting agreements, all the things to run a business. But then you also have your IP counsel, who's going to be helping you file these patents and to prosecute that. And I'll defer that until later because I won't cover intellectual property. But that's also a concern as you're thinking about capital is a lot of that early IP you file is so important, but that's also when you have the least amount of money. And you're trying to balance all of those factors as you're filing your provisional applications and then moving that forward. But the IP, one of the expressions in biotech is it's all about people and patents. And so the patents are important, but you're trying to get a lot of that groundwork done when you don't have a lot of money. So it depends on, you know, if it's technology spinning out of a university, you know, hopefully that IP is really good, that they put together a good package. But then as the biotech company continues to develop their own IP, you know, a lot of times you have your medicinal chemists writing your IP, and then you'll send it to the patent attorneys. They'll look at it just a little bit, but you're trying to keep the billing rates down, and you're playing that game to survive. So all of that then, you got the IND in, and as I said, the FDA has 30 days to review it. If you don't hear from them in 30 days, you can start, but they can come back to you at any time and put you on clinical hold, but you can't go earlier than 30 days. So you submit it, you don't hear anything, on the 31st day you go, and then we can go to, and then this is kind of, now that we're in clinical trials, it's really referred to as development. So now you're going to start your clinical studies in humans. And you're going to start in phase 1, and as I talked about last week, your phase 1 is usually, but not always, in healthy volunteers. And I said, typically it's younger adults, college students looking for beer money, that kind of thing. But if you have a toxic regimen, or you have some sort of antipsychotic, if it's not ethical to give that type of compound to someone who's healthy, sometimes you will do your phase 1 work right in patients. But usually, the dosing time frame is not long enough to really get any true efficacy rate. But sometimes you will go right into patients. These are prospective studies in the sense that we're not generating data and then retrospectively looking at that data and seeing what it says. It's forward-looking. Study participants are followed from some well-defined point. Baseline is usually, sometimes it might be when they get initially screened, but from baseline. And these trials are divided typically into 3 big buckets, as we talked about last week. Your phase 1, where the focus is really on safety. Phase 2, where you're getting your proof of concept, does it work in the patient population of interest? And that's kind of hypothesis generating. And then your phase 3 is where you're testing the hypothesis. These are your pivotal studies that you do for approval. Sometimes you'll come across the concept of phase 4 clinical trials, and these are really studies that are going to be done post-approval. One reason you may see these studies is they're looking for long-term safety of a drug. You have a drug that's going to be used chronically, much longer than what you tested in phase 3. Then you may have to do a phase 4 study and just be following patients for years. And sometimes there's also phase 4 studies done for marketing purposes as well. And also during phase 1, you're doing all your non-clinical work to get ready for phase 2. And so this deals with your manufacturing. You're going to be scaling up to larger and larger scales so you can produce more and more of the drug, and it still has to be the same quality, if not better quality. You're going to be doing longer-term toxicology studies. So now you're going to be doing toxicology in 2 species for a longer period of time to support the longer dosing that you're going to see in phase 2. And then you also have regulatory activities underway as you're getting ready for further interactions with the agency. So we can go to the next slide. It says we talked about phase 1. This is where you're looking at a small group of healthy volunteers. The emphasis really here is on safety. If you are in patients, you may get some, but it'll be very little efficacy data. This is all about safety. The objective is really looking at PK and PD.
So PK is pharmacokinetics. It's the blood profile concentration in the blood. Pharmacodynamics is the action of the drug. And its effects, both good and bad.
Yeah, exactly. And then you're also getting an idea here of your maximum tolerated dose. Where are you seeing toxicity at what levels? And this gets important too. You have some idea of what you think the therapeutic dose is going to be, and then you want to know how much window do you have in there before you have unacceptable toxicity. So this is where you start getting a feel for what that could be. It's a very controlled process. It starts with single ascending dose studies, SAD studies. If that looks good, you move on to multiple ascending dose studies where you start exposing patients for a longer period of time. And I have some examples of what these studies look like. And there's some other studies that go along here as well. You might be doing food effect studies to see what happens if you take it with food or without food. For some reasons, depending on the indication you're going for, you may also go into special populations in phase 1. Usually just a single dose as well, but to the extent these patients are healthier within these categories, you may look at hepatic or renal impairment patients, geriatric, things of that nature. Or if there's something you saw in your preclinical package that you need to explore further in terms of safety, like, for instance, drug-drug interaction, you may have some studies to look at that as well. In a typical phase 1 program, you're looking at between 50 and 150 volunteers. This stage of development will take about a year, 2 years, and can cost anywhere between $3 and $10 million to complete. And so, as we were talking about, you've already done 3 years of work through early discovery to IND-enabling tox, and this will take another year or 2. So now you could be about 5 years out from the initial time you had an idea, you had a target, and you got a lead. So in looking at a typical, and there's exceptions to all of these examples, but this is kind of like what a standard phase 1 SAD study could look like. You're going to randomize the patients. It'll be placebo-controlled, so there will be a placebo in the study. It's a single ascending dose, focuses really just on safety. You may be looking at healthy male and female volunteers, a range of doses. You may start at 20 milligrams and slowly work your way up. Smaller studies, 8 subjects per dose. You may have 6 on drug, 2 on placebo. You'll do 1 day of dosing, follow up for 7 days. If that looks clean, then you move on to the next level. So it's a very structured design, and it's all about safety. After that, you'll move on to your MAD study. It's very similar, except you'll have a smaller grouping of doses, still randomized, still placebo-controlled. A little bit larger, maybe 10 subjects per arm. You still have the placebo, but here you're going to be looking at 14 days of dosing. So you just kind of keep working your way up. So subjects may take 10 milligrams for 14 days. You'll follow them up for 7 days. If it looks clean, a committee will meet to look at the data. You get the thumbs up, and then you move on to the next one, and then you move on to the next one. And a lot of times these are just done in a single center, and they're pretty straightforward. So then in terms of phase 2, so now assuming your phase 1 looked clean, everything looked good, now you're going to move into your patient population of interest. And this is where you're going to be looking at both safety and a preliminary look at efficacy. You're going to evaluate whether the drug has biological activity in the patient population of interest. This may or may not be with what you're going to test in phase 3. So for instance, you may have an endpoint in your pivotal studies that the patient has to be on drug for years. So instead of doing that in your phase 2, you may be looking at different biological markers as a surrogate for that efficacy, but you are looking at efficacy either directly or through a surrogate. You want to start taking an idea of what your adverse events are, laboratory abnormalities, any safety issues, patients are being dosed for longer. And the design of this phase 2 is to do 2 things. You want to start narrowing down that optimal dose. So a lot of times in phase 2, you'll still take a couple doses in because you don't know what the best dose is yet. And it's going to be heavily influenced by what you've seen in your phase 1 as well as all the preclinical work that you've been doing. So these studies are much larger. Now you're moving into multiple centers, 100 to 300 patients. And these studies will also take a couple years to complete. And they will cost between $10 to $40 million as well. Some phase 2 studies are pretty small. In areas of oncology, you might do a small phase 2 study. But in most indications, these are going to be much larger, more expensive and time-consuming studies. You're going to need a pretty robust toxicology package in place prior to exposing this many patients to drug for a long duration. You might need repro-tox. Could be before phase 3, but you might see it here. And you're going to be doing longer-term repeat dose studies. So you've selected your 2 species, and now you may be doing 3-month, 6-month studies in rats, mice, or in dogs prior to being able to initiate your phase 2 study. And those cost several $1 million each to do. And then following a successful phase 2 program, here's another opportunity to interact with the agency. You'll have an opportunity to meet with the FDA for an end of phase 2 meeting.
Sore subject.
Yeah, sort of subject. But, and this is where you can get guidance and input on the phase 3 program with the FDA. And they look at all the data you have with your packages, and you start getting their input into your phase 3 program before you embark on what is a very long and expensive program.
Good question. You were saying that for biotechs, they tend to have like this kind of molecule that's roughly good.
Mhm.
It could be better, but it's safe. If the timescales we're talking about here in a couple of years, if they have an improvement that they want to introduce, do they have to go back to start over at phase 1? How much wiggle room are they?
Yeah, and so if it's a brand new molecular entity, you are starting the whole process again. So you're always kind of looking at the data that you have in your backups, the data that you have in your lead. If the backup is a big leap forward and it's only 6 months behind, you may kill the lead and bring the backup forward. And so you're always looking at that. So you're looking at the data that's being generated by your lead compound, and you're looking at what you have in your backups, and sure, you may switch them. And so it's not uncommon for a small biotech, given the capital constraints, to be pushing a compound forward, partner with Big Pharma. Big Pharma looks at it, yeah, we don't like the lead, we're going to kill the lead, we want to bring one of the backups forward. But then you do have to go through that whole process, but you do it in a more informed way because you have all the information that you have from the lead. But yeah, you're just playing with that balancing game and what you're saying.
What happens if those patients, they agree that you participate, but then for some reason they disappear or they don't want to do it anymore and there's like missing data?
Then there's ways that the statisticians have to deal with that missing data, but it does cause problems. And so, you hopefully you have really good investigators who run the studies, who are, they know how to do it, they're very comfortable with doing it, and it's a risk. And so when you power a study and you think about a study, you have to think about a certain dropout rate. And you have to kind of power that in. Otherwise, yeah, if you get a dropout rate that exceeds what you were expecting, and you've already closed enrollment, then the power of your study and your statistics, do get damaged. And so it is an issue. And so the more toxic the regimen might be, the more complicated the dosing regimen is, the longer amount of time it is on drug, it becomes more difficult. Okay. So an example of a phase 2 study, so you're randomized, placebo-controlled. Here's your proof of concept. So you're going into patients and you're doing your proof of concept. You're assessing safety, tolerability, but this time you're also assessing efficacy. You're taking in your patient population of interest. An example here is for some disease state, you're targeting more mild to moderate patients. Your final doses are going to be based on what you saw in your SAD and MAD studies. And so depending on what you saw, you're going to determine your optimal doses for phase 2. In this example, I'm assuming that you're taking 2 doses in. You haven't chosen a single dose. You're taking 2 doses in. So you've got 3 arms, 2 for the 2 active and 1 placebo. This example is 6 months of dosing with a 14-day follow-up, so it's a much longer study. And now you start going into multiple centers. So you may have 38 centers running this protocol and running this study. And so it becomes quite a bit to manage. So once you start moving, as a small biotech, once you start moving up into running phase 2 studies, you start building a big clinical sciences group. So you probably have a chief medical officer who's a doc who's running the study. That person may have a VP of clinical sciences, so that's another doc who's more hands-on. And you've got, maybe 4 to 5 clinical scientist folks, clinical research associates, who are managing the centers and managing the studies. And you also have a CRO who's running them. So it becomes a very big process. And,
Contract research, clinical organizations, you can contract out your clinical trials to, increasingly people do that, to these organizations.
companies like Quintiles, this is what they do. They run clinical trials. And so you'll select a CRO that best fits, the area that you're working in, and you'll work with them. So you're paying them money to run the study, but you also have a team that's helping to oversee them and is helping to oversee the sites. Because once you get this thing kicked off and once it gets going, as you had mentioned and you brought up, you don't want to mess it up. You don't want to have a situation where you have a really high dropout rate. You don't want a situation where the sites mix up drug and placebo. It can happen. And then you got to start dealing with all those problems and all that data. And if you spent tens of millions of dollars and you're a year or two down the road, it can become a big problem. So these trials are watch very, very closely, not only from a safety perspective, but also from just an execution perspective, and make sure you get the numbers and the data that you need.
I don't want to interrupt you. I have to go to another meeting, but I wanted to tell everyone how grateful I am to James. These are invaluable talks. Invariably, I learned some stuff. And so, and also want to reassure you for your projects, we will be giving you help. I met with a couple of you during office hours. There are ways to assess all this. We don't expect you to be a full-blown venture capitalist investor by the end of this, but it's just a learning experience. So James and I, or Chris and I, or somebody will be meeting with you after our tutorial sessions in the projects if you need help. But once again, thank you, James. As you know, he's an Air Force pilot. Well, I call you a pilot. Okay.
Does anyone have any quick questions for Phyllis regarding the project? Yeah.
Regarding companies being US or abroad.
I think they can be abroad. Yeah, that would be interesting. It's pretty flexible and a lot of fun. And don't worry. This is really, as I explained to Brian, for example, this is really about learning something that you're not going to get in the rest of the university. So right now, it's a fire hose to the mouth. Don't worry, this stuff will start to come on. It takes years and a lot of effort to learn it. So we'll get as much of it into you as possible. Okay, any other questions? Thanks, James. See you tomorrow.
See you tomorrow. And so a lot of times in phase 2, you talk about your primary endpoint, safety and tolerability, but you really are also looking at efficacy here, which may be your approvable clinical endpoint, but if it's not reasonable to look at that in a smaller, less duration study, you'll start looking at biomarkers and surrogates for that. So then you move on. Oh.
Just curious, is it possible if you taking your compound abroad and work with so you take your compound to Europe and you work with the French regulatory agency and you get phase 1, phase 2 data, can you use that data for the FDA?
Yeah, you can definitely submit it, and depending on kind of what you did and where you want to go, you may have to repeat a few studies or this and that, but the data is not completely lost. You can definitely bring that data in. And there's more and more effort underway of trying to harmonize regulatory in Japan, regulatory in Europe, regulatory in the US, and trying to get that more streamlined so you're not trying to do different things in different places. But even as you start into phase 2, a lot of times, you got 30 to 40 centers, you'll have centers in the US and Europe. And I've been involved in companies where we've done our phase 1 work in Australia, just because it was, for whatever reason, it was a little bit easier to get into Australia, maybe less requirements, but you still go through, committees to approve it, and you're still at, hospitals. But sometimes you can do phase 1 work in Australia, then bring that data to the FDA, and open up an IND, with a lot of that data. So yeah, it's not lost. So now, assuming everything's been successful through phase 2, you had a successful phase 2 study, now you're going to move on to phase 3, which a lot of times we refer to as your pivotal studies. These are your registration studies. These are the studies that form the basis for your application to the FDA for approval. And so these are large-scale efficacy and safety studies in a substantial number of patients, and you're really getting a much better view of what this compound is in the clinical setting than you did in your phase 2. So your phase 2, you talk about as being hypothesis generating. Phase 3 is where you're going to test that hypothesis. And these are large studies where you can extrapolate those results to the broader population. And this is what you're doing for approval. And this is why they're pivotal studies. Usually, and this is becoming more the case, I think now, is you're going to do 2 confirmatory studies. And so you're going to do 2 large studies, usually with the exact same protocol. There may be some differences. One study may have an extra dose just to show a dose relationship, but essentially what you're going to be doing is 2 large studies, and both studies have to be positive to get approval. In the past, and with some exceptions now, sometimes you can do one even larger single study, and some of the statistics are different. They may be looking at a p-value of 0.01 instead of 0.05 in the two studies. But the best way to think about this traditionally is you're going to do two large independent studies, and they both have to be positive. These programs could be 900 to over 2,000 patients. They can take 3 to 4 years to complete. And if you've got a large phase 3 program, it can easily run over $100 million. And so you want to, so all the work you've done leading up to this is getting the right compound, selecting the right dose regimen, and then getting all your CMC, all your toxicology, and you've done all this work so you can pull the trigger on a $100 million plus investment that's going to take 3 or 4 years to find out what the results are. So big bets with long time horizons until you figure out what the answer is. In the background of all this, your CMC work is continuing. You're scaling up to higher and higher amounts of being able to produce drug product. And your toxicology package to initiate phase 3 could have required long-term carcinogenicity studies. Could be a phase 4 commitment, but if they wanted it phase 3, now you're looking at 2-year toxicology studies in 2 species. Very expensive studies. Additional, you could be looking at reprotox studies, and then anything else that they may be seeing. Anything that's coming out in earlier preclinical studies, anything that might be showing up in the clinic, you're going to drive down on it with additional toxicology studies. So they're very expensive as well. And these costs and all this work is independent of the $100 million I'm talking about for the clinical study itself.
We talked to FDA.
Well, you've talked to the FDA at your end of phase 2 meeting. So you've gotten all your phase 2 data, you have all your CMC information, all your preclinical, all your tox, and you bring that to the FDA and you have that discussion prior to starting this. You have your end of phase 2 meeting. And now the FDA will give you guidance, and you can choose to agree with all of it, or, I mean, there's got to be safety parameters, but you may decide, I don't want to quite do it the way the FDA said. And you do that at your own risk, because you're going to spend a lot of money and it's going to be a long time, and you can go back to the FDA and say, we told you that you should do X, Y, and Z, and you do it. But yeah, you should be interacting with the FDA before you do this. And when you interact with the FDA, as a small biotech company gets bigger, you will have a regulatory department whose job is to take care of regulatory. Usually small departments, 3 or 4 folks. But you'll also hire consultants to help you as you prepare for these milestones. So you're hiring consultants who've worked for the FDA, who maybe headed up the division of the FDA that you're going to go present to. But yeah, you do a tremendous amount of regulatory work. You really have to incorporate that feedback. You have to be smart before you pull the trigger on this.
I mean, after phase 3, basically, you're
Hopefully. That's the goal. And then I'll cover kind of I'll cover the NDA and what you might hear back from the FDA in the subsequent slides.
So when you hire consultants or hire people that have worked at the FDA to consult, to basically guide this process, do you have to declare any sorts of conflicts of interest or things like that?
There's probably some, and we've worked with consultants who work for the FDA, so I'm sure that's between them and the FDA, and I'm sure they would have that cut off. But a lot of times, they might be able to join you in those meetings, and so it's transparent.
Okay.
It's nothing behind the scenes, but yeah, a lot of times before you do these things, you will look to bring additional consultants in. So to file an IND, your in-house regulatory department is probably just going to crank that out, no problem. End of phase 2 meeting, you might start looking at bringing in some outside consultants here and there to help you with that as well.
Are they typically outside, consultants that have, just their own business is just them, or are there consulting firms that have, a whole suite of people?
In my experience, it's been a lot of individuals. Hiring a lot of individuals. You may have people who headed up regulatory at Genentech for years. Now they're kind of semi-retired, so on the side they'll do some of that work. So a lot of times you have very senior regulatory folks. Maybe didn't work for the FDA, but they've been doing regulatory for 25 years. And they're not working for a company now, and they're doing consulting. So depending on the complexity of what you have, at times you will bring in consultants to help you understand that. So what could a phase 3 study look like? As always, randomized, placebo-controlled. Your pivotal studies in the same example, I've been using mild to moderate patients. Here it's more in the clinical setting, so they may be on background therapy, whatever the standard of care is, they're on it as well. You'll probably have 2 arms Here, you're not looking at dose ranging anymore. You're going to have what you think your target therapeutic dose regimen is, and you're going to have a placebo. Large study could be 700 patients per arm, so you can have 1,400 patients in each study, 2 studies. You have almost 3,000 patients that you're tracking. Not uncommon in a large study like this. You can have 100 centers in the U.S. for one study and 100 centers in the U.S. and Europe, excuse me. So you're managing 200 sites in the U.S. and Europe. And here your primary endpoint is efficacy, and you're also looking at safety and other endpoints, anything that you should be looking at, anything the FDA is also giving you guidance that you should look at. So these are big studies, big commitment of funds. And then once you've gone through all of that, then you get to the NDA or the BLA. And if all of that's been successful, and this has been 7, 8, 10-year process, and could be easily over $100 million spent, then you can prepare to file your new drug application, your NDA, that is required for marketing a small molecule in the United States. If it's a biologic that you've been taken through this process, and outside of the manufacturing and some of the early kind of drug discovery work that I outlined, a lot of this is the same for biologics. The approach to the clinical studies, having the toxicology packages, having the CMC packages together, all of that's the same. The end of phase 2 meeting, the checkpoints with the regulatory agencies, all of that's the same. Where the differences may come in is obviously scaling up a biologic is a lot different than a small molecule. I don't have any personal experience with that. And then some of the early drug discovery, you're not running thousands and thousands of compounds through a screening cascade when you're trying to get an antibody that has the right specificity and affinity. That process is a little bit different. I'm not too familiar with it. But in overall, it really is the same thing. Prior to filing the NDA, you can have a pre-NDA meeting with the FDA. So here's another opportunity for you to interact with the agency and get guidance on preparing that package that you're going to submit. So you're going to do that after the phase 3 data, prior to you submitting the NDA. This is an integrated multi-volume document. So it's got all your preclinical work, all your clinical work, all your CMC work, 300 to 500 volumes. This could be 200,000 pages of information that you file electronically with the FDA. And this could take you 6 or 9 months to put together. So once you get that positive data and it looks good and the clock is ticking, you put a big team on it, and it still takes 6 or 9 months to get it out the door. The NDA is organized into several modules: administrative, kind of overview, your quality, so your CMC, which is really your manufacturing package. Because at this point in time, you're manufacturing this at a very, very large scale. You're using a lot of drug product because you're trying to satisfy patients all over the world. Your safety, your clinical study reports. And in 2012, if you're filing an application with the FDA and you're asking them to look at clinical data, so basically the type of application I've outlined here, the fee for them to look at it is about $1.8 million, and that pays for them to have a team of people review the application. So after you've submitted it, the FDA has 45 days to inspect the document to ensure everything is in order. If so, then they will file the NDA, or if not, they may request that there's more information or better information is required, and they will refuse to file until those changes are made. So there's a 45-day process where they review it, and then if it's good to file, then it will go through several layers of review by various experts. And so it's medical, pharmacology, chemistry, statistics. It's a very comprehensive review. If it's a situation where there's a high unmet medical need, the FDA calls it a therapeutic advance. So say this is a really important cancer treatment, the FDA may grant priority review. Otherwise, they're going to grant a standard review. The most recent timelines I've seen from the FDA, so looking at approvals from 2006 to 2010, standard review took 396 days, and a priority review took 183 days. So getting the priority review done in just about 6 months, and a standard review is taking over a year, on average. Could be a little shorter, could take longer. Throughout this process of them reviewing, there's kind of a back and forth that could be going on with the agency, where they're asking questions and clarifying that. And then at the end of all of that, they will issue either you got approved, or they will issue what's called a complete response letter. And what the complete response letter is going to tell you that the initial review of the application is complete, and it cannot be approved in its present form. And that can mean any number of things. Maybe there's some additional preclinical work that you need to get done before they can approve, or it can mean you got to run more clinical studies before they can approve it.
What percentage of people that do file this NDA are, what percentage go through and what percentage don't? I mean, you've obviously committed such a huge amount of money and you've even paid a big fee to the FDA. What's the attrition rate?
You know, on average, when you look at some of the kind of the benchmarks overall, I've seen between kind of 90-95% approval rate, somewhere in that range. And obviously, you as a company, you have a lot to do with that. If you try to, I don't want to say garbage, but if you don't have a compelling package and you're hoping to just cram it through and you're hoping that there's an unmet need out there and this will somehow get through, you know, you're taking that risk on you. If you have a real tight, good package, you've had good interactions with the FDA, you know, you have a much higher probability of getting approval. But just because you submit, just because you had what you thought was a positive phase 3 study and you submit, doesn't mean it's 100% that you're going to get approved. And when you get a complete response letter, it's not good, and depending on what they want, it can be very, very bad.
I have a question about the clinical trials.
Mm-hmm.
So, from what I noticed, all the subjects are healthy individuals or mild to moderate cases.
In my example, yeah.
Yeah. So there are cases where like serious like patients for in serious conditions might.
Oh, absolutely, if that's your indication. And so these I use just as an example, but there's so many different flavors of them. Depending on what your target indication is and where you want to go. If you're going into very severe patients, maybe they're refractory to all types of cancer treatments, you're going to, you know, kind of the train wreck patients, and that's what you would study. Absolutely. So you're studying, especially in your phase 3, you're studying the patient population you want approval for. So this, in just this example, I have just mild to moderate, but absolutely. And so from there, if that's kind of a real overview of fire hose of the whole drug development process. And so as you look at companies and you can see where they are in their pipeline, you can kind of get a feel for what have they accomplished and what is still ahead of them and how much time that might take. And there's exceptions to all of this. But especially if you can get you know, reports, as I talked about last week, from equity analysts who cover that space. So I think you might be able to get here at Stanford. You know, they'll start doing some of those projections that the study may take a couple of years to complete. You can also look at clinicaltrials.gov and find the studies on the website, and many times they'll have, when they anticipate to have completion dates. You can look at that and see when you'd expect them to finish. And so from there, I'll move into an overview of intellectual property, and I'll pause to see if there's any questions before I move in that direction. Okay. So in talking about IP, you know, kind of important to kind of talk about right up front is it's not a positive right in the sense that you have an issued patent and now you can do something that nobody else can do. It really is a negative right in that it excludes others. I have this patent, and because it's mine, you can't do X, Y, and Z. In terms of getting patentability, so your ability to get a patent, the first thing you look at is it novel? So for a new invention, there must be one difference between you and what's called the prior art, what else is already out there. For the most part, this is a pretty low hurdle. It just needs to be a little different than what's already out there. The more significant hurdle comes in the next step, and that's non-obviousness. So there needs to be some level of unexpected benefit. And this can't be a logical step by someone skilled in the art. So someone who's skilled in whatever art it is, antibodies, whatever target, or whatever it might be, for someone who's skilled in that art, what you're submitting can't be the logical next step. So this is a little subjective, but the takeaway is there needs to be a significant gap between what you've done and what everybody else has done already to make it patentable. There needs to be a written description and enablement, so you're going to have to reduce it to writing, and so you need to describe it well enough so that someone who's skilled in the art could practice it. And a fourth topic is really it has to be patentable subject matter. You know, for a lot of what you're looking at for the companies for your project, they're going to be looking at patentable subject matter, so this isn't an issue. But things like cloned human beings are not patentable in the U.S. Embryonic stem cells are not patentable subject matter in Europe. So clearly, if you're trying to get a patent on something that's not patentable subject matter, you're not even going to get through that first gate. Something important to note, too, is having a patent doesn't necessarily mean you have freedom to operate. And so when if you're in a small biotech and you're going to be doing a licensing deal with Big Pharma, at some point in time they're going to do their IP diligence on what you have. And one thing they're going to look for that's not up here is chain of title, which is just that they want to make sure that you as the small biotech have rights to what you want to license to us and what we're going to pay for. So if you spun technology out of Stanford, they're going to go back and make sure you truly have a license to that. And a professor doesn't have a license to it as well, and you didn't know about that. So they want to make sure that you have chain of title. Then they're going to look at patentability. So to the extent you don't have a patent issued yet, they're going to have to make their judgment on the likelihood that a patent will be issued. But in addition to that, they're going to have to do what's called a freedom to operate analysis. And as venture capitalists, when we make investments, we go through those things as well. And a freedom to operate is where you can start spending a lot of money. Because at this point in time, what you're looking at is you're trying to look at the entire landscape of patents out there and try to determine that even if you get a patent, someone else doesn't have a patent that somehow supersedes you and blocks you from practicing with your patent. So in the small molecule world, you might have a patent on a compound, which we would call a species, but somewhere out there there's a genus claim, a higher claim that blocks it. And so those analyses are very expensive. You go with a law firm, their patent agencies start doing various searches to try to determine, do you have freedom to operate or not? And that's something you can't really do in-house as a biotech company, is your freedom to operate analysis. Types of small molecule patents you could get. Composition of matter is what everybody looks for. That's where you're actually getting a patent for the exact chemical structure. Formulation patent is the formulation you're using. When you think about kind of formulation, that kind of takes you back to non-obviousness. So a formulation patent is one avenue you might use for life cycle management. So you get your composition of matter patent. When that patent is starting to expire and generic competition comes in, traditionally Big Pharma will say, okay, we're going to reformulate that, maybe go into a controlled release, change the formulation somehow, try to extend that patent life. And it's a good strategy, but in order to get that patent, it's also going to have to be non-obvious. So if there's 5 salts out there to choose from to reformulate your compound, and you choose one of those 5 salts, that's really a logical next step in the process, and you probably won't get that patent. But you could look at doing a formulation, changing that formulation. If it's novel, non-obvious, that can give you a patent as well. So you hear people talk about having formulation patents. And also method of use patents. So you can get a patent for using your compound, say, in Alzheimer's disease. And then if you can then later on do clinical work in Parkinson's disease, then you can get an indication there. Then you can get a method of use in Parkinson's disease. But one thing to note in that strategy is a physician can go ahead and prescribe the generic drug anyway. And so if you have the exact same dosage strength, the exact same dosing regimen, and your indication in Alzheimer's goes generic, Watson and various generic companies come out with their generics, a physician can still prescribe the drug for Parkinson's and they can fill it with the generic. And so as you're trying to get method of use patents, you're also going to want to try to change the dosage form, the dosing schedule, in order to try to protect that. And so that's one of the strategies in life cycle management. In order to kind of bridge this to antibodies and proteins, more in the biotech space, patent strategies for, say, antibodies could include patents for antibodies to a novel antigen, patents to antibodies to a novel epitope. You could be looking at antibodies with novel properties, whether that's affinity or specificity or some sort of activation or neutralization. Excuse me. You could actually also be looking at patenting specific antibodies, kind of like the composition of matter. Here is the exact antibody. I'm patenting that antibody, and not just the antigen that I mentioned before. But the antigen is a very good, because a lot of times if you're really novel, you'll combine the two. You're going to get a patent on your actual antibody, but then you're also going to get IP on that antigen. You're going to block anybody from that antigen, even if they have a different antibody. And then mixtures of antibodies or polyclonal antibodies. So if you're looking at antibodies or antibody-like companies as part of your project, those are the patent strategies for that too. So it mirrors what you see in small molecules in many ways as well. So in terms of the timelines and the costs for this is an eye chart. I'll just kind of walk you through and just kind of point out the real high-level things. So in terms of your intellectual property, kind of the timelines and the costs, to mention right off the bat, these costs are probably, should be viewed as more like the floor and not the ceiling. This is kind of the minimum you would pay to have this done. So the first thing you do is you file your provisional. This is your priority document. Kind of in the beginning, you file as broad as you can. You're going to refine it over the year that you have. You can file multiple provisionals, and you can just kind of continue to file during that year to strengthen what you have. But this is kind of where you get that line in the sand. You file your first application, but it's a balancing act because it's just as good as what you put into it. Kind of like garbage in, garbage out. So if you're rushing in to try to get an early filing date, but you don't do a really good job, then you got to kind of live with that. So this is where you start. You file your provisional. Your outside legal fees could be, $12,000 to $20,000 at the really at the floor. And this doesn't include all the work that you're doing internally. So your chemists and stuff are probably writing up all this IP work, and they're putting a lot of time and effort into that when they're not at the bench doing something else. And you're looking at about 20 grand at least to get that done. So as I was talking about earlier, this is a stage when you have very little capital, and you're really trying to conserve how much you spend on IP attorneys, but sometimes that's the investment that you have to make. So you file that provisional, then at that point you're going to have a year until your what's called your non-provisional filing or your US or PCT filing. And really what this is this is where you are filing the patent, and what you file gets set in stone. So this is where you've worked for another year, you kind of really thought about your claims, and this is the patent that gets set, and that's it, and this is what's going to get reviewed. And two things that's important to note here. One is 6 months after this date, so 18 months after you filed your provisional, it goes into the public realm. So prior to that, what you filed is not available to the public. But at a minimum of 18 months, and it could be, usually it's 18 months and a couple weeks, what you filed now becomes available to the public. So others are going to have a chance to see what it is you're working on at that point. And that's also where your 20-year patent term starts. It starts on your non-provisional filing. That's when you have 20 years from that date until you have expiration. Just at a real high level, there's kind of two ways that gets extended. The first is with a patent term adjustment. And this is a system that takes account into delays that take place in the review process between the applicant and the patent office. And there's complicated formulas for how all that works out, but at the end of the day, if you get a patent approved, you may get a certain number of days extended to the life of that 20 years. Many patents are approved with 0, but you can maybe get 10 days, 20 days, 30 days. Sometimes it can be significant, but it's usually smaller. The other way could be extended, and that's for, I should mention, the entire patent. The other way is a patent term extension, a PTE, and this is a process that takes into account delays between the applicant and the FDA in the review process. And it'll apply to a single patent. I believe some sort of PTA is seen outside of the US as well, but I think the US is the only one that has the concept of a PTE extension. So you file your non-provisional, starts the 20-year clock. That'll also cost around $20,000 to do that. And then at that point, especially if you file, most people now just file a PCT, which is a patent cooperation treaty, gives you the ability to then have that patent go out to other countries within the PCT, and there's like over 100 countries in the PCT. So you don't have to keep filing specifically in each jurisdiction. There's a process in place to allow you to facilitate that. So at any time after you file your non-provisional, but to a maximum of 1.5 years, you have to decide which countries you want to actually file in. And that's what we call our national phase. And usually for a small biotech company who's capital constrained, you're going to file in at least 5 countries. You're going to file 5 to 6 countries. You're going to file US, Europe, Japan, Canada, Australia, and China, usually. And that can cost you around $80,000 to $100,000 to do that. So this is a very expensive kind of time in your patent prosecution when you go into the national phase. If you want to file real broadly, say you have a compound that you feel is going to be a global partnership with pharma, this is the next real big thing, you may decide to go into a total of 12 to 13 countries. And now you start opening up to your BRIC countries, I mean, not in here would be Russia, but Brazil, Mexico, India, South Korea, Singapore. Russia should be included in that as well. And this can easily cost over $200,000 or more. And most of the expense is not coming from redoing the claims and all of that, because everything's already set in stone. There's no more drafting anymore. This is really around foreign associate fees and translations and things of that nature. But as a small biotech, when you get into this phase, you got to balance how many countries do I file in versus how much money do I actually have at my disposal.
So, does the national jurisdiction apply to, say, pharmaceutical that came out of it in Mexico, but the company is actually based in the U.S., but they produce it in Mexico, but you only file in the U.S. for those products?
And this is really about, so the patents block people from commercializing the products. They don't block people from doing research on them. So, once patents get into, once patents publish, so for instance, if you're working in an area, I don't know, you're working in Parkinson's disease, and you're starting to generate compounds, you can look into the patents of competitors that are published. You can see what Merck is doing, you can see what Pfizer's doing, and you can go ahead and synthesize those compounds. You can do that in your lab, and you can test them in your systems to see how your compounds compare. There's nothing to block that. But once you have IP in a country, you're blocking someone from selling it commercially.
Does it stop people from doing it?
Yeah, you can't sell it. Yeah, you can't. So if you have IP issued in the U.S., someone can't manufacture it in Mexico and then try to sell it in the U.S. That's what they're blocked from. They can manufacture it in Mexico and go sell it in Singapore if you don't have any IP in Singapore. If they go through whatever regulatory process they need to have approval in Singapore, they can do that. You're not blocking people globally, but you will block people in the countries that you file. And so you're trying to look at the potential of that compound and kind of weigh that against how much is it going to cost me to file in all these jurisdictions. And then what happens is once you've done that, then you have at least 1 to 2 years where you're waiting for your examination to begin. And then once that examination begins, then it can be 1 to 3 years until the patent actually gets approved and issued. And that could cost anywhere between $25,000 to $80,000 a year. Depends on how many office actions. It really depends on what is going on with the patent agency at that point in time in order to try to get it approved. And also after this, there's maintenance fees, annuity fees. There's other fees that you're paying to various patent agencies to keep the patent alive. And so, we kind of walk through the drug development process where you're looking at an 8 to 11-year process that costs well over $100 million. Here you're also looking at another very expensive, long process that's going on right alongside all of that. And you don't want to get too far ahead of yourself where you're starting to spend a lot of money in clinical studies only to have a patent that can't be issued. So you're running all of these things, and you've got to balance all of these risks and all of these capital constraints all at the same time. So that's why, as a small biotech company, when you're going into really big areas, we're going to have to run real big clinical studies, and you're going to go into areas where the IP is a minefield. There's a lot of people playing in this space. You partner with big pharma because you're just not going to be able to do it yourself, and you're going to need to bring that resource on.